Now that we know a little bit about carbon, let's talk about some of the large carbon-based molecules that we find living organisms using on a regular basis. There are four major classes of biological molecules. You have your carbohydrates, your lipids, your proteins. All of these you should be familiar with from your diet, at least in a general sense. And then we have nucleic acids. So our molecules that are encoding genetic information and passing on genetic information and using that information to produce functional organisms. There's a really nice summary chart on page 70 of your textbook that lists some of the basic characteristics of each of these molecules, the types of bonds that they produce, their monomers or the components that make them up. And for each one of these, you will need to know all of those things. You'll need to know the basic, most basic monomer and the type of structure that those monomers take. You'll need to know the bonds that link the individual pieces of these large molecules. And you'll need to know characteristics of those monomers, specific characteristics such as functional groups and things like that. There's a lot of memorizing in this chapter, so you will need to take a little bit of time, memorize some things about these molecules. And this chapter is really important because it's sort of a foundation for a lot of our other chapters. It's a foundation for our chapters on DNA structure and replication and producing proteins, and also our chapters on photosynthesis and cellular respiration, where the body is using carbohydrates to release energy, or a primary producer such as a plant is actually storing energy in those carbohydrate molecules. So this chapter is really important, but it's also a chapter that tends to trip up students, and that's because most students have not had much experience with the complicated chemistry of these large biological molecules. I'm not quite so worried about understanding all of the details of the chemistry, but I do want you to know why these molecules are important and how they're put together. And so let's start talking about carbohydrates. So carbohydrates are carbon and hydrogen based molecules. You can see carbo and hydrate in the name there, and they're basically sugars, either simple sugars, individual molecules, or very complex sugars that are linked together into these long carbohydrate molecules. So you should be familiar with this in your diet. Carbohydrates are in things like bread and pasta, candy, sweet drinks. Anytime you add a sweetener, you're adding a carbohydrate. All carbohydrates are going to be made up of a single monomer, or a single part, called a monosaccharide. Mono means one, saccharide means sugar. So a monosaccharide is your most basic sugar structure. And they are really the building blocks for much more complicated sugar molecules that the body uses for storage of energy and production of energy. Your monosaccharides always come in multiples of sort of a single carbon unit. That's a carbon and an oxygen and two hydrogens. So if you look at glucose there, glucose is C6H12O2, which means it's made up of 6 carbon molecules, 12 hydrogen, and 2 oxygen. All of your monosaccharides are going to be a multiple of that basic CHO structure. So monosaccharides can be found in a linear form or in a ring form. If you look there, we have on the left, we have a variety of complexity of our monosaccharides. On the far left, you have triose, which is a 3-carbon sugar. You can see it's made up of 3 carbons, a couple of oxygens, and some hydrogens. You have very specific functional groups in sugars. You very often have those hydroxyl groups and that carbonyl group, which is the carbon double bonded to an oxygen, which makes these molecules polar. Remember, oxygen is electronegative, and so when there's oxygen in a structure, it tends to make the whole molecule polar. This is important because carbohydrates like to interact with water, which is really important when they are being utilized in the body. You can also see some more complicated sugars down there. You've got glucose, which is a 6-carbon sugar. That's going to be sort of our main focus in this chapter, is glucose as sort of your most basic monosaccharide, your most basic carbohydrate. We also have ribose, which is the sugar found in RNA, and we'll talk about that a little bit later. Monosaccharides are more often found in a ring structure, and that's because the ring is more stable. And so they often will, the linear structures, if they are linear, will fold into a ring structure. And then the ring has oxygen and hydrogens coming off of it, sort of depicted here as hydrogens and oxygen above and below the ring structure. The ring structure is not flat, but it is generally drawn that way for simplicity. So monosaccharides, and specifically glucose, are the major nutrients for the cell. Glucose is the basis for the processes of cellular respiration and photosynthesis. So in photosynthesis, plants take in sunlight energy and convert it into chemical energy. And that's a really important process because it stores and maintains, or basically stores, all of the energy that living organisms on Earth need. So photosynthesis takes in sunlight, converts it or stores it as glucose. And then in the process of cellular respiration, that glucose is broken down. So plants will, after it stores the energy as glucose, will then break down glucose as needed to produce energy for cellular work. Or other living organisms will eat those plants, ingest that glucose, and then break it down themselves. So we'll really get into the details of photosynthesis and respiration in chapters 7 and 8. But just keep in mind that the reason that we're studying these molecules is because they are important later, and they are important for the most basic processes that living organisms perform. Once you start linking your monosaccharides, you begin to produce much more complex molecules. And most of the carbohydrates that are utilized by living organisms are actually more complicated than just a single carbon ring, than just a single monosaccharide. So living organisms, particularly for storage of some of these molecules, when we're talking about carbohydrates and lipids, will produce chains of these molecules, and that, again, that's related to its function, and we'll talk about that in a minute. When you link two monosaccharides together, you get a disaccharide, saccharide sugar, so 2-sugar molecule, and they are bound by something called a glycosidic linkage. So here's one of our words that we need to know. Carbohydrates are linked by a glycosidic linkage. So you can see in our picture on the right, you have glucose and fructose. When you link them together, you produce a covalent bond between those two rings, and now you have the molecule sucrose. So sucrose is a common sugar in our diet. They are linked by something called a dehydration reaction. So we saw this earlier where you remove a water molecule from the two molecules, remove the components of a water molecule, I should say, and produce a water molecule. And what you have left is a covalent bond, and in this case these two sugars are linked by that oxygen. So that's a characteristic of a glycosidic linkage. You have a oxygen sort of linking those two molecules. So disaccharides are used a lot in cells. But carbohydrates will often also form polysaccharides. So poly means many, and these molecules can be huge. Our carbohydrates, proteins, and DNA all produce these really large polymers that are just linked repeats of their most basic units. So polysaccharides can be hundreds or thousands of monomers long, so they make these massive structures. Some of the carbon structures can be really complicated. But remember, this always goes back to structure and function. We have some very specific carbon structures that have very specific functions in the organisms, and that function and that structure is determined by the specific monomers that that carbohydrate is made of, and also the position of those glycosidic bonds. So if you look at that ring, depending on the ring structures, depending on how the rings are positioned and where the bond actually forms, you can have different functions for these carbohydrates. So here we have a couple of examples of the different types of carbohydrates. We have two major functions of carbohydrates. One is storage, so storing carbohydrates, usually to be broken down later for energy. And then we also have structural carbohydrates. So I really like this image. This is from our book, and it's a single image that shows a variety of different carbohydrate functions in this one scene. So we have a farmer here holding potatoes. We can see there's plants in the pictures, there's potatoes, there's muscle fiber, and it just shows some of the ways that carbohydrates are used by living organisms. So the first structure we have is starch. So starch is a storage protein in plants. So it's a way that plants store carbohydrates for energy. So if you look at that structure, we can see the glucose monomers. They're sort of in the middle of the screen, and they are linked by that glycosidic linkage. You can see that little oxygen linking those rings together. And if you look at this starch, it's not very highly branched. You can actually have branches coming off some of these glucose molecules, and that just allows for a little bit more compact storage. But as a plant needs to use this starch, it'll begin to break off those monomers and be able to break down those individual glucose monomers and release the energy that's being stored in their bonds. So plants store their energy as carbohydrates. They, in this case, store it as starch. And it's just one of the ways that plants store their energy. So after photosynthesis, they produce a precursor to glucose. It is converted into glucose and then linked into these large starch molecules. In animals, some of our energy is stored as carbohydrates. And in animals, the carbohydrate that is used for energy storage is called glycogen. So if you look, this structure is very similar to the starch above it, but it's much more highly branched, and that's to make it a little bit more compact in the cell than the starch is in plants, and that's because we move. We need to have a sort of compact energy source. Starches are not nearly as compact as fats, though, and we'll talk about fats in a little bit. We also have our plant cell, and here we have an example of a structural carbohydrate. So carbohydrates are used for energy storage, but they're also used for structure because polymers of carbohydrates can make some very strong structural support molecules. So the plant cell wall is made up of a carbohydrate called cellulose. So cellulose is fiber in our diet. It's something that cannot be digested by mammals, and it's the substance basically in our diet that sort of keeps our digestive tract moving. But cellulose is made up of glucose molecules just like the other two, but the position of the ring is slightly different. If you look, we have an alternating up and down facing glucose ring in that cellulose. And again, structure reflects function. When the rings are alternating, it lines up the carbohydrate rings so that you can get some hydrogen bonding forming between polysaccharides that make up this cellulose fiber. So you essentially get a carbohydrate rope. So when you look at glycogen or starch, there's not really any interaction, any hydrogen bonding going on between the different strands of carbohydrates. But then when we get down to cellulose, you not only have the covalent bonds that link those rings, but you also have hydrogen bonding in between. And remember, hydrogen bonds are individually weak, but collectively very strong. And so you get these interactions between those cellulose microfibrils and sort of this really strong, really dense structure. And that's why cellulose can support a plant. The function of the plant cell wall is to give support against gravity, and it is able to do that because of those hydrogen bonds between the cellulose microfibrils. Another structural carbohydrate is chitin. So chitin has a more complicated ring structure you can see down on the bottom, but we see the alternating position of the rings there, and then we have some different functional groups. But chitin also makes a really hard structure, and it's found on the outside of organisms that have exoskeletons. So the support structures on the outside of these organisms, very tough molecule. So carbohydrates are used for energy storage, but lipids are also used for energy. Lipids are a category of a molecule, biological molecule, that is really important for living organisms, but they're not polymers. The other molecules in this section are all polymers, so repeating units linked together. Lipids are not, but they're still important in our diet, and they still share some characteristics with these other large biological molecules. So you guys are probably all familiar with lipids, even if you're not familiar with the word. All of the fats in our diet fall into the lipid category, and they're really important because, again, they are the compact energy storage that animals use. So lipids are a diverse group of molecules. There are a variety of different basic forms of lipids, and they can be small or large, but they're really not true polymers. They're not extremely large, complicated molecules. One characteristic that unites the lipids is that they are hydrophobic, and we'll see why that is in a minute. But they are hydrophobic, which means they don't interact well with water, whereas our other molecules, our carbohydrates, proteins, and DNA, will all interact with water, and that's really important in living organisms. So let's talk about these three different types. Our first type of lipids are fats. You should all be familiar with fat from diet. We find fat in the foods we eat all the time. It's in meat, it's in fats are the main component in different types of oil and foods that have high fat content, things like butter and dairy products. So fats are not a large polymer. Fats are made up of basically two units, one that repeats and one other unit. So they are put together by a dehydration reaction. So there we see a little bit of a similarity with our other large molecules. The two main components of fats are glycerol and fatty acid tail. So the glycerol molecule is a very simple sugar. We've got the carbon, hydrogen, and oxygen there. Very simple sugar. And a fatty acid is a long chain of carbon and hydrogen. So highlighted in yellow there, we have this hydrocarbon chain. And if you recall from earlier in chapter 3, we talked about hydrocarbons, and we said that one of the characteristics of hydrocarbons is that they're very high energy. There's a lot of energy stored in those carbon-carbon bonds and in those chains. And so fats are really important in animals because they are the main energy storage molecule. They're very high energy and they're very compact compared to carbohydrates. So a fat is a glycerol attached to 3 fatty acid tails. So each of those fatty acid tails is attached by a dehydration reaction. Remember, we have the loss of water to add the one molecule to another. And that dehydration reaction produces an ester linkage. So the ester linkage is highlighted there. It is characterized by a carbon double bonded to an oxygen and then another oxygen linking it to the glycerol backbone there of that molecule. So this is called a triacylglycerol because it has 3 fatty acids attached to that glycerol. Now this is a single fat molecule. This is not a repeating polymer like in our carbohydrates. And the cell will store these individual molecules, but they're not linked to be any bigger than that. There are 2 types of fats. And the first is saturated fat. The other is unsaturated. Again, you're probably familiar with these terms, but let's talk about what that means in terms of the actual chemistry of these molecules. In a saturated fat, you have no carbon-carbon double bonds in that fatty acid tail. So one of the characteristics of the the saturated fats is that they pack really, really tightly. And because of that, they tend to be solid at room temperature. So we have butter as an example here. Butter is a saturated fat. And those fatty acid tails essentially form a straight line. And so if you think about those very highly ordered molecules stacking on top of one another, you get these really dense molecules packing together. So you get a lot of compact storage, and that's why animal fats tend to be solid at room temperature. So if you think about bacon grease, you cook bacon in a pan, you let the pan cool, that bacon grease will solidify. Same thing with butter and just some other fats in our diet. The saturated fats are the ones that tend to clog arteries and things like that when you have a diet that's really high in saturated fats because they pack together, because they are solid at higher temperatures. They can line arteries and things and become really dense in those areas, and that's when you tend to have heart problems and things like that because those saturated fats can build up in those places. Our other type of fat are the unsaturated fats. So these are often thought of as sort of your healthy fats. And the unsaturated means that there's a carbon-carbon double bond in at least one of those fatty acid tails. The saturation refers to whether or not all the carbons are completely surrounded by hydrogen. And when you have a carbon-carbon double bond, you have each of those carbons has one less hydrogen, so it is unsaturated by hydrogen. And the reason that these are more healthy is that because of those kinks in the tail that are produced by those carbon double bonds, you don't get that really tight packing that you can imagine in the saturated fats. And so these unsaturated fats tend to be liquid at room temperature. So these are going to be your oils. Our example up there is olive oil. Others are canola oil, vegetable oil, things like that. There are some oils that are actually solid at room temperature. Coconut oil is one. But in general, your unsaturated fats are going to be your oils because they can't pack tightly. So it takes a much lower temperature before they start to solidify. Which is why they tend to be more healthy in your diet. They are less likely to pack together in your arteries and things and clog them up. There are 2 different types of unsaturated fat. One is cis, the other is trans. And you're, again, probably familiar with trans fats as sort of a buzzword in nutrition right now. If you remember from our discussion on isomers, cis means on the same side of a double bond. So this image here shows a cis fat where you have the kink that produces or produced from the carbon-carbon double bond, and the groups are on the same or the cis side of that double bond. Trans fats don't occur naturally, and trans fats are an artifact of processing saturated fats into unsaturated fats, and that's to change the temperature that they're solid. And so during the processing of saturated fats, in an attempt to make these cis fats, a byproduct is also the production of some trans fats. And the problem with trans fats is that your body doesn't know how to deal with them. And so it can cause a lot of health problems because they don't occur naturally and the body doesn't have a way to efficiently break them down. It's often recommended that people eat a diet that is high in unsaturated fats in order to sort of reduce those problems that you get from eating a diet high in saturated. Our next type of lipid is sort of a modified fat called a phospholipid. So phospholipids are really important in cells. Phospholipids are the main component of the outside layer of almost all living cells. The outside layer of a cell is produced by something called a phospholipid bilayer. So there's a picture down there on the bottom. The structure of the phospholipid is a little bit different from the other fats that we saw. Remember, the other fats, regular fats, are produced by a glycerol bound to 3 fatty acids. The phospholipid is only bound to 2 fatty acids, and instead of being bound to a third, it's bound to a phosphate group instead. So that's where we get the phospho part of the lipid. And this molecule is really important because it has 2 different properties within the same molecule. One is a hydrophilic head, which it allows it to interact with water on the outside or the inside of the cell, and then it has hydrophobic tails. And so this phospholipid bilayer that is produced basically produces a barrier between the cell and the environment inside and the environment outside, with those hydrophilic heads pointed out and the hydrophobic tails pointed in. And those hydrophobic tails limit the movement of things in and out of the cell. So it's an effective barrier in keeping just random molecules and things from moving in and out of the cell. And when we get to chapter 5, we'll talk a little bit more about this phospholipid bilayer and some of the characteristics of it, why it's so important. Our last type of molecule are the steroids. So steroids are hormone precursors. So they are a lipid because they are hydrophobic, but they are a very different structure than either the fats or the phospholipids. Steroids are made up of 4 fused rings, and they differ by their chemical groups. So cholesterol is a steroid that is a precursor to estradiol and testosterone, are sort of sex hormones that help to differentiate male and female characteristics. And you can see these molecules are really, really similar, but they differ in their functional groups. So cholesterol from the diet is modified into these different hormones based on the functional groups that are attached to those those structures.